So, hey, welcome to Unknowns. It's a day one in phytomicrobiology. We did a small experiment to study the room bacteria or study the bacteria in the airborne bacteria and fungi. And we use something called plate technique to understand what kind of bacteria. This is very important because once you start your pure culturing and characterization, you need to know who is living around you and how it is living, how fast it is floating, how fast it is settling down. That's why this is called settle plate technique. This is very important technique because they are used in various industries. Okay, pharmaceutical, food and beverage, hospitals, biotechnology, cosmetic, personal care, many more industries. In pharmaceutical industry, for example, they are used for clean room validation and monitoring. They are used for aseptic filling area. Even in the good manufacturing practice, they have settle plate as a first technique. If you are a technician and you walk into pharmaceutical industry, usually this is the first thing they will do. They will do the settle plate. They will study the colonies very next day. Every day they will maintain a document what is been happening, how this will give you an idea of how clean your room is. In food and beverage industry, if you see quality assurance in processing zones, hazardous analysis of critical control points, HACCP, they need to have this settled play technique to have an idea. In hospitals, for infection control, for getting an overview of nosocomial infections, they need to do settled play to assess the bacterial diversity. Even in biotechnology and R&D labs, there is something called DNA, RNA work zones, and they will continuously monitor for are they maintaining a biosafety standards. In cosmetic industry, for even in the packaging area, the microbial spoilages should be studied for which settle plate is important. Even for ISO certification, ISO 22716, in the cosmetic industry, they will demand this monitoring way. Even heating ventilation systems in other processing units, they need to have this as a control check. It's a performance checks of HVAC systems. And even whether the HEPA filter in the room is working fine or not, you need to validate it by settle plate technique. In dairy and fermentation, you will see milk processing, cheese aging, fermentation tanks, everything should be regularly monitored. Even for regulatory and certification compliances, for example, ISO 14698, routine environmental monitoring is important. If you're applying for that, better have 3 years data ready before applying. So I thought we will do an assessment of the room we are going to use for phytomicrobiology. So this is very easy technique, low cost, non-instrumental, easy to implement. That's why we took a challenge. The interns took a challenge of assessing this. Yesterday, while we did our day one, we put a plate in two different places. These are those two plates. It's in nutrient agar plates. We tried to study the unique morphotypes that is being suspended in the air. We identified this unique morphotype in green color here in table one. There are 4 tables. We used 2 tables. We open the plate and let it open for 20 minutes. And these are some EPS producing colonies which are not there in table 1. And this swarming colony is not there in table 2. So this shows there is a heterogeneous distribution of bacteria in the air. We did not stop here. We tried to compare the colony morphology. We used AI to compare this. And the table 1 have few pinpointed colonies which is missing in table 2. And the wrinkled colonies, more extracellular polysaccharide secreting colonies are found in table 2. And swarming colonies 1 is been found in table 1. So altogether, there are 8 unique morphotypes. Okay. Altogether, probably 3 are very unique. So we may have a 5 common morphotypes across the combining both the plates, but sorry, in every plate, in both the plates, we may have 8 unique morphotypes. Okay, so the colony count is 60 colonies in table 1 and 100 colonies in table 2, meaning the density of bacteria that is being distributed in the air is also different. We try to do a settling rate assessment where this is a formula for settling rate where number of colonies are actually multiplied by divided by surface area of the spindle plate. We used 9 centimeter diameter Petri plate. The top cap is 10 centimeter diameter. The bottom one is 9 centimeter diameter. We expose the plate for openly for 20 minutes, and we want the unit in meter square per hour. That's why we have 10 power minus 4, because we want to convert this surface area of that is 9 centimeter into meter square. So we have n into 10 power 4 divided by a into t. This is a formula, and this is a step-by-step calculation. We found that in plate 1 had 60 colonies and 100 colonies. When you apply that, so plate 1 had around, the settling rate is 472 colony forming units per meter square per hour. In plate 2, it is 786 colony forming unit per meter square per hour. Okay, so this is how the settle rate of the particular ambient should be calculated. See, this settle plate is very easy technique. Even if you want to assess the bacterial concentration of any lockdown spaces, when you unlock a particular space, if you want to assess whether the space is safe or not, even selective media can be used for assessing. That can be used for diagnosing a presence of particular pathogen in the air also. So this is an interesting results we got. The internship continues.